Okay, just to get things started, we drew some turkeys. If you're watching this, yes, this is AP Bio. We drew turkeys. Show you all the. And now we're back. We will do a nature walk later. All right. So, looking now at lycophytes and more vascular tissues, and of course we're going to start to look at xylem and phloem more now. But this is where vascular plants start to dominate the natural landscape. And when we say vascular, we are talking about xylem and phloem. Xylem conducts water and dissolves minerals up from the roots. So that's what actually, it's like the veins, the blood vessels for the plant. And phloem conducts sucrose and other organic compounds throughout the plant. So this is kind of the liquidy matrix that flows throughout the xylem. And then lignin, this is what strengthens the cell walls of conducting cells in xylem. And I think of lignin as synonymous in animals as being sort of like collagen. Collagen is the strong fibers that make up our vein and arterial walls. And so lignin is like that for plants. Most seedless vascular plants are homosporous. That means they have one spore, single spored. And they're wind-blown spores and are dispersal agents. So this is how they're going to use spores to spread or procreate. Now, Cooksonia is an early vascular plant, and there are some fossils of Cooksonia, and it looks kind of like a branched tree. And of course, if I drew this, it's going to look awful, but I'm going to give it a shot. Cooksonia sort of branches off on these spore-forming structures and looks something, and there are pictures of this in your book. And I am not doing it justice, I know. No, not really. But we do have those fossils actually in Michigan. So looking further at lycophytes, the roots come off of branching sections and are undergrown rhizomes. So what I was trying to draw was those branching structures with rhizomes at the bottom. But the roots evolved as lower extensions of the stem. So the roots sort of developed from the stem, which a lot of times we think of roots as being the beginning, right? Not really. They're an outshoot of the beginning of the plant. And so leaves are what are called microphyls and have only one strand of vascular tissue, and most likely evolved as simple side extensions of the stem. And that's what I was trying to show you, is those microphyls coming off of each branch. There's a microphyll, and there's a microphyll, and this would also be growing down as well. And microphyls bearing what are called sporangia, also called sporophylls. And they're grouped into club-shaped strobili. And the sporophyte generation is the dominant structure. So again, that sporophyte is like the plant or the tree. Okay? And yes, we will take a nature walk after we cover sporophytes and flowering plants. Okay? This is not wildlife. We can only do one nature walk a year. What? We'll see. Yes. What? Yes, sir.
Are you going to turn the plant dissections today?
No, we will not turn in the plant dissections until we cover flowering plants. Okay? So better than my drawing could possibly be, of course. You see the branch structures. Here you see the microphyls that we talked about, and extending off of them the strobili. On the strobili, you have the sporophyll and the sporangia attached to that. This is where the spore comes from. On the actual leaves themselves, those branched leaves, you can see that you have the vascular tissue in the middle, the xylem and phloem, where the fluid comes from. And this is just the stomata, where the opening on the leaflet actually is to exchange gas. Down here, if you look at the vascular tissue closely, you see the xylem is in the center with the phloem surrounding it. Again, the xylem where the fluid transfers through, and the phloem, that liquidy matrix that allows circulation in the plant. And then growing down, you have the rhizome. I know my E got extended out. I don't know why these are doing that when they come in. But the arterial stem, called a rhizome, is going to stem downward and create the outshoot or the root. Now, in today's legumes, we still have sort of an early foot or a rhizome ancestor of this, where they actually house a symbiote, a bacteria, which fixes the nitrogen for the plant. We talk a little bit about that in regular biology. We will get into that more, but that's part of the nitrogen cycle. And this plant is part of the beginning relationships of plants with those particular types of bacteria at these what are called rhizomes. Now looking at pteridophytes, that's a fun one, and megaphils. The pteridophytes, these include ferns and their allies. How many have seen ferns in here? Walked through the woods, or who's gotten flowers from a boy or a girl? Who's seen flowers in a bouquet? Oh my Lord. All right. So it is Monday, back from break. But that sort of decorative green foliage is usually a type of fern. And what do you see on the back of the fern a lot of times?
Good job, Joe.
You don't see bumps, little egg things that look like eggs? I need to take you guys out into the woods. Oh my word. Yeah, you're killing me now. So on the back of ferns, often you will see these little, they're actually the spores that are about to be released. But a lot of times I used to think, I'm sorry, when I first saw them, that they were eggs, like there was an insect laying eggs on the backs of these things. But it's sort of how they reproduce. Okay. We'll talk more about that. What? I can tell. You guys are like, we don't know anything, Mr. Mason. Take us out in the woods, but you probably do. We might have to just make a trip to the state park in the spring. I will be taking you guys ice fishing.
Yeah, that sounds fun. Do you have to bring our own fishing pole?
I would suggest it because the stuff they give you there is, it's like tying a rope to a stick.
My sister dropped my fishing pole in the middle of the lake. I was mad.
So the dominant form showed up in the late Devonian period through the Carboniferous period. The cool thing about the Devonian period is what? What is special about the Devonian period? Let's see if you guys know this. What's special about the Devonian period? What is special about the Devonian period? See what we're coming up with. See what you guys came up with here. Devon was born. Things died. High oxygen. Well, some of these are true. I cannot draw the UP.
The bridge was built?
So, it's not that old. All right. So this is our period, right? This is where Michigan is. This is what we're carved down the Devonian period. I know that's not that special, but it kind of is to geologists. So at any rate, I think that's something we should all know, the period that our state exists in, as far as where our fossil record is carved down to. It's also why we don't find dinosaur fossils here, because they're all pushed out west by the glaciers. So we have the Devonian period is where we're looking at, up through the Carboniferous period. And of course, the Carboniferous period is where you have that explosion of plant life, right? It's where all our fossil fuels come from. So back to pteridophytes, they have what are called megaphils. These are broader leaves. They allow plants to efficiently collect solar energy, allowing the production of more food and possibly more offspring. How are we doing? All right. Now, where are we at? Where's your note? You get out your little writer there and put some words on that in the blanks. There you go. So this resulted in increased fitness for plants. What is the structure inside of plants that allows this to occur. Let's see how much you guys remember. Be specific. What is the structure inside of plant cells that allows this to occur? The collection of solar energy. Oh, my Lord. I pray that you know this. Be specific. The more specific, the more right you will be. There's a general answer, which everybody in here should know, and then there's even more specifics that you could add to this. You could tell me the type of membrane that I'm talking about, not just the organelle that I'm referring to. They look like stacks of pancakes, right? What are they? No, whatever that was. They are in the chloroplast. Yeah, the thylakoid membrane, right? The stacks of thylakoid pancakes. Yay. So remember, you have those stacks of thylakoids, which have the light collecting units called, what are those light collecting units called that are on the thylakoid membrane? Chlorophyll, yep, chlorophylls, they're pigments, right? Photosystems, photosystem 1 and 2. Whoops. So on microphylls versus megaphylls, our microphylls, they're going to have a smaller single strand of vascular tissue. That's why they're called microphylls. The megaphylls will have a large, broad, leaf-like structure that will have vascularization through branches. And you'll actually see the veins beginning on the leaves, okay, that stem from that xylem and phloem layer in the center of that stem. So I tried to draw this earlier with little success, but you get the idea of the branch stem system, where one begins to dominate the other as they extend outward, eventually the other one becoming vestigial, but still feeding the outer layer. Here we are looking at a ginkgo structure. And then, of course, there are horsetails. Oh, I'm really going to test your history. Here's a history question. You guys like history. What did they used to use horsetails for? When I say they, I mean Native Americans. Toothbrush. Eric? Smoke signal. Let's go. Q-tips, sofa Q-tips. So, actually, there's a lot of silica in them. Oops, sorry, I went the wrong way. There's a lot of silica, which is glass. It's almost like fiberglass, so they actually use them to scrub their dishes. Did you say scrub? I'm sorry. Yeah, so they were like the early scrubbers, so to speak. And if you ever grabbed a hold of one and tried to run it across your hand, you get the idea. It like grabs a hold of you a little bit. It's almost like grip paper. But they used to use them to clean dishes. Well, kind of, yeah. So they consist of one genus, the Equisetum, and approximately 25 species of seedless vascular plants. They inhabit wet, marshy environments. So we find these. If you go down to the beach, who has seen these on like the edge of the bay? You guys probably don't even know what I'm talking about here, but they're these like skinny things. They got like little sections, and if you grab hold of them, they actually kind of are rough. They're really rough, actually. Like if you have to pull them out, they might actually hurt your hand if you didn't grab hold of them tight enough. The rhizome produces tall aerial stems. So if you pull out these cattails, if you pull out these horsetails, you'll actually see this rhizome structure on the base. And if we get to go to the state park, I know we shouldn't pull them out because it's probably against state park rules, but Maybe we'll like accidentally step on one and knock it out of the ground. Whoops. But you get the idea.
That's on YouTube demonstration.
What? I will. They contain whorls of slender green branches, and leaves are small and form whorls at the joints. Oh, I can't believe you guys haven't seen horsetails. These are right here in town. There's probably more of them here because of the marshes. You guys have seen these, right? Yeah, right there. That's about what they're going to look like. You guys have seen these, right? Yeah, that's horsetails. Okay? They are everywhere. Oh, I went the wrong place. Oh, now I'm just reading for interest. I'll come back to that later. Okay. So the horsetail will grow in these sections, and then there will be these, at each little node, there will be whorled structures or leaves. Okay? Now, there will be the nodes in between, and depending on if they are, which stage of development they're in, you'll either see these branches or you will not. This time of year, they're probably all falling off. You're just going to see the long segmented stalks with maybe the strombilus on the end. Now, if you did pull them out, don't pull them out because they are good for the bay. They prevent erosion and things like that. But if you were to pull them out, you would find the rhizome at the root. And to think how prehistoric, we're talking back to the Devonian time where these things came from. So these are older than dinosaurs. So in a way, you look at plants, and they're way cooler than finding like T-Rex running around. No? Well, maybe not.
What does it really do? I mean, they're not really active.
They make oxygen. I'm kind of glad they were. You know. What did T-Rex do for you? Bite your head off?
They work actively.
I see. Okay. Is it? Oh, okay. So then looking at whisk ferns. So these can exist as epiphytes that plants live basically in the crux of the trees. What do you, tell me something about epiphytes. Tell me something about epiphytes. Yeah. I don't care what. Just tell me something about epiphytes. You guys have probably learned about epiphytes since you were in elementary school, right? Where do you find a lot of epiphytes at? Yeah. Since I started talking about horsetails, I've had the DuckTales song in my head, just so you guys know. Is that terrible? Did you guys watch DuckTales when you were kids? No? I'm old. No. What are you guys doing? So a lot of epiphytes in the rainforest, for sure. So, oh, you just got back from Florida. You saw a lot of epiphytes, didn't you? Yeah. So did you drive or did you fly? Okay. Well, there's a ton of it. They call it Spanish moss. It grows all over the trees. It kills trees, right? You see that stuff on all the trees? Yeah. No. If you go look at any palm, any tree in Florida, they're going to have epiphytes growing on them. So epiphytes are kind of cool because they actually can draw in the moisture from the air around them. They don't need a root structure in the ground. So there's two basic genres here, the Soleatum and the Thymocypterus. Now the Soleatum species, they lack leaves, they have horizontal rhizome that gives rise to an aerial stem that forks repeatedly, and the sporangia are on short side branches. The Thymocypterus, species have appendages resembling reduced megaphylls. So they often look as like this, almost like there's a pile of yarn, but it's a plant. And a lot of times they're just in the crux of a tree or where a stem branches off. And a lot of times you find them in the palm trees or in the fig trees in Florida. That's why I brought up Florida, because I see a lot of them whenever I go there. Did I go too fast? I'll go back. I'll go back to the Davinian period. I don't. I did not say that. That's something that some other teacher might say, but not this guy. I can't say that out loud. The river Styx. Doesn't the Mackinac Bridge go across the river Styx? Only on Wednesday. That was kind of an inside-outside joke. Mostly because rivers are outside. So, oh lordy. Now looking at ferns, we've seen a lot of ferns, even though you guys won't admit to it. You see a lot of ferns, whether it's in the flowers that get delivered or hopefully in a walk in the woods. But there's approximately 11,000 species of ferns. They're most abundant in warm, moist, tropical regions, although in Michigan, because we have so many wetlands, we actually have quite a few ferns. Large conspicuous megaphylls are what are called fronds, and those fronds are often what they put in with your flowers when you send a bouquet to a loved one. And then they're divided into leaflets, and then the dominant sporophyte produces windblown spores. That's why I said if you look at the back of a fern, you'll see things that look like an insect laid eggs on them. That's actually the reproductive portion of the fern. Who's flipped a fern over and seen the bubbles on the back of it and wants to admit it? Yeah, we've all. Okay, all right. Seems really. I'm freaking out that you guys haven't noticed this. By the way, it's like I don't. All right. So. All right. All right, we need to go on a nature walk. I just need to take you guys up north and we'll just go out in the woods and go for a walk. We don't need to go in. Perhaps. So this is the sporophytic structure. The large, this is where you normally notice, this is where you look and you say, oh, it's a fern. But if we go back down to the fiddlehead, I think this is a fun structure just because it's called fiddlehead. I used to think, you guys, I used to think these were a completely different plant. I used to go, oh, that's a really cool plant. Look at that. I wonder what that is. And I would look for it in my plant book, and I couldn't find it. Why couldn't I find it? Because it was actually the fern at a very early stage. I'm not even kidding you. Like in college, I'd take botany, and I wanted to know what that stinking structure was, this right here, this fiddlehead. And I didn't learn this in high school, and so I got my book out, and I had to do this big herbarium where you collected and flattened and dichotomously keyed all these plants out. And I collected this, and I tried to key it out, and I couldn't find it. And then, like 2 months later in lecture, they talked about fiddleheads, and I was like, oh, that was what I was looking at. And I didn't realize it was a fern. So the fiddlehead is that early structure. This will actually unravel and grow into this large sporophyte. Okay? And, of course, we have our rhizomes, and then we have roots that branch off as well. Now, remember, the rhizome is like that branching structure that connects each fern to the other one. And so what are you saying, Mr. Mason, that these are all the same fern? Yeah. Okay? Which is kind of weird because you look at a fern forest and it's all the same thing. Like giant clones, it's like a clone army of ferns. Right? The aspen, yeah. So the largest organism, here's something really cool. The largest organism in the world is in the Upper Peninsula. It's a popple tree. It's an aspen. It is the entire span of the UP. It's all genetically the same. Isn't that creepy? You think? I mean, it is a little bit, you know. Maybe not. It has rhizomes that run. If you don't believe me, walk up to the edge of a popple forest and dig, and literally you'll be able to pull up the rhizome and connect one tree to the other. They're all the same. What? If you set, well, they all can live independently and keep going, but yeah. So no, you won't. It's not live, die, repeat. You know. Yes. Yeah. You separate them, then it'd be like, yeah. I don't know. Now we're taking this way too deep. So on the bottom of those leaves, this is where I was saying it looks like eggs, but in a way it kind of is, right? It's a spore, it's sporangium. Okay. So on the bottom of the leaf, you have these things that look like eggs, and they are the sporangium that hold what are called sori, and this is where your spores are going to be released from. And as we continue the cycle, we have what is called the annulus, which is where the sporangium is going to be at. Yeah, it does a little bit. And now it's going to get too small, I know that. After you develop spores, that are going to be formed through meiosis. And remember, meiosis is where you cut your genetic information in half, which means that germination is going to occur next, where it starts to grow. Now, this is important because we have that alternating of generations. We go from being 2N to being 1N, or haploid. I've got to get this off. I can't get down here. Okay. At any rate, this starts to grow, and this is still 1N. So this 1N structure is going to divide through mitosis to create this kind of a multicellular haploid individual. It's the gametophyte. And the rhizoids will start to develop here. So it's already starting to branch out at a very early phase. Okay, the antheritis is going to form and release sperm. And once that sperm finds an egg in the archegonium of another structure, it's going to then be 2N, right, through fertilization. And then it will return into that diploid structure and grow into a larger structure through mitosis and start to create the fiddlehead and so on, and eventually the sporophytic fern. So the fern is a good example of that alternation of generations, and we mentioned that in biology as the alternating between 2N and 1N over and over again. So what do we use ferns for? Well, to decorate, right? We talked about that. We like to send flowers to girls or boys or whatever. You never know, right? So some ferns are edible and they're used as a food source. The azolla harbors nitrogen fixation. We talked more about the rhizome in legumes of today. They're the early ancestors are the ferns. The cyanobacteria N is growing in rice paddies where it fertilizes the rice plants. So if you go up into the mountains of China, you find these fern forests that surround all of the steppe farms of their rice paddies. And the farmers actually grow these for two reasons. They prevent erosion and they fix nitrogen. And the ferns and other allies are used as medicines in China. What? Yes. Actually, China's. I mean, you think of China as being the city, but the wilder parts, like the mountains of China, are nothing like what we think in our brains as China being. Oh, and they're allies. Yeah, I know. Right? Yeah. Yes, they do. And extracts from ferns have also been used to kill insects. And they're used as decoration, of course, when we send flowers to people. So there are a lot of ferns in mountainous regions because China has so many mountains. You find a lot of ferns there. Yes? Oh, okay. You guys ever watch Wild China on Netflix? Not that one. No. I've seen that too. Who's that guy? Kurt Russell? Kurt Russell? Yeah. That is a funny movie. The green-eyed Chinese lady? That was the one? Yeah. No. All right, we'll stop there. We'll take a break.